Hello everyone, welcome to my channel Dr. Tapati's presentation. Myself Dr. Tapati Vanjade.
Today, topic of the presentation is multi-enzyme complexes and multifunctional enzymes. I'm going to define multi-enzyme complexes and multifunctional enzyme, substrate channeling, advantages of substrate channeling, and then I'm going to give you a few examples of multi-enzyme complexes and multifunctional enzymes like fatty acid synthase, pyruvate dehydrogenase, 2-keto dehydrogenase, proteasome, and few other examples. Kindly stay with me till end of this video for the complete understanding of this topic. Multi-enzyme complexes and multifunctional enzymes catalyze consecutive reactions of a metabolic pathway. Enzymes are subunits of multi-enzyme complexes, and multifunctional enzymes are organized in such a manner so that the product of one becomes the substrate for an adjacent enzyme or enzyme subunits. They allow for a tighter regulation and a more rapid and efficient response to changes in equilibrium between substrate supply and demand than would the individual enzymes. Metabolic reactions that are linked by multi-enzyme complexes and multifunctional enzymes have clear advantages over reactions that are catalyzed by individual or unlinked enzymes. They allow to segregate certain biochemical pathways into one place in the cell. Such complexes could be important modes of regulation. Let's start in detailed discussion on multi-enzyme complexes and multifunctional enzymes. Multi-enzyme complexes are aggregates of several copies of one or several enzymes packed into one assembly that catalyze a single or a series of sequential biochemical reactions in the same pathway taking place in the cells. The complex is held together by non-covalent forces and may easily undergo dissociation and reassociation. Each component of the complex has own Km for substrate, cofactor, Vmax, and pH optimum. The assembly of cooperating enzymes into multi-catalytic complexes, also known as metabolons, which has become a well-accepted concept in cellular metabolism, at least in principle. Let's know about multifunctional enzymes. A multifunctional enzyme is one that contains two or more different catalytic centers on a single polypeptide chain. It shows different enzymatic functions in a single polypeptide. Whereas in my last video, I mentioned that in case of multi-enzyme complexes, those are aggregates of several copies of one or several enzymes or polypeptide chains packed into one assembly, and that assembly catalyzes a single or a series of sequential biochemical reactions. That means main difference between multifunctional enzyme and multi-enzyme complexes is that in case of multifunctional enzyme, a single polypeptide chain shows multiple functions or multiple functions, whereas in case of multi-enzyme complexes, several enzymes or several polypeptide chains packed into one assembly, and then they catalyze a single or a series of sequential biochemical reactions. Multifunctional enzyme complexes are proteins created through the joining of two or more genes that originally encoded for independent proteins. They can reach molecular masses of medial Dantos or MD and above and exceed the ribosome in size. That means molecular size is very large. Covalently attached swinging arms such as lipoyl groups, biotinyl groups, and phosphopentathionine groups are essential to their action mechanisms. The existence of multifunctional enzymes and multi-enzymatic complexes that catalyze consecutive reactions of a metabolic pathway is sometimes accompanied by important functional consequences for enzymatic regulation or enzymatic activity regulation. Let's see a few examples of multifunctional enzyme. Fatty acid synthase is A very good example of multifunctional enzyme. Fatty acid synthase is a key enzymatic complex in fatty acid synthesis from acetyl-CoA and malonyl-CoA in the presence of inadequate pH into long-chain saturated fatty acids, and it is expressed at high levels in liver and adipose tissues but at low levels in other tissues. Fatty acid synthase is not a single enzyme but a whole enzymatic system composed of two identical 272 kilodalton multifunctional polypeptides with 7 functional domains, each known as Ks, At, Mt, Dh, Er, Kr, and ACP, in which substrates are passed from one functional domain to the next. Its main function is to catalyze the synthesis of permitted. Full forms of functional domains are ketoacyl synthase, that is for Ks. Then AT means acetyl transacylase, MT means malonyl transacylase, DH means dehydrogenase, ER means enoyl reductase, KR means ketoacyl reductase, and ACP means acyl carrier protein. Along with these 7 units, thioesterase is also involved in this fatty acid synthesis process. Second example of multifunctional enzyme is reverse transcriptase, which is also a multifunctional enzyme. That means it has RNA and DNA dependent DNA polymerase activity as well as ribonuclease H activity, and is responsible for the reverse transcription of retroviral single-stranded RNA into double-stranded DNA. Third example is glutathione ACE transferases, which are multifunctional enzymes involved in the metabolism of a broad variety of xenobiotics and endogenous compounds, including antibiotics, steroids, prostaglandins, and leukotrienes, and are abundant in the liver. They are involved both with bilirubin transport and detoxification of bilirubin, the toxic catabolite of heme, which is bound and thereby neutralized. Fourth example is CD38, which is a small glycoprotein and a multifunctional enzyme located on the plasma membrane and inner nuclear membrane. Its expression generally correlates with poorer outcomes in a number of hematologic malignancies. The major enzymatic activity of CD38 is the hydrolysis of NAD+. Sirtuins are NAD+ dependent enzymes. Therefore, there is a link between CD38 and sirtuin activities. Let's compare dissociated multi-enzyme system and multi-enzyme complex. Many enzymes in living cells catalyze chains of reaction in a sequential order, either in a biosynthetic or a catabolic pathway. In its simplest form, the enzymes of such a system remain free in the cytosol as independent entities, each interacting with its own substrate, which is also present in the cytosol. The product formed from each reaction is liberated and is acted upon by another enzyme of the sequence. Intermediates diffuse away. In this figure, you can see that S is the substrate, P is the final product, where in this sequential reaction, E1, E2, E3, and E4, these 4 enzymes are involved, and A, B, and C, these are intermediates of this reaction series. They diffuse away in the bulk solvent. Whereas some of the multi-enzyme systems may operate in a different way, when the enzymes are closely associated with each other to form a multi-enzyme complex. Intermediates in the reaction sequence catalyzed by an enzyme complex are covalently bound and do not diffuse away from the complex. Not only are the enzymes bound in the complex, but also the intermediates of the enzymatic pathway remain bound to it, and only the final product is released. Let's know what is metabolite channeling or substrate channeling. Metabolic channeling or substrate channeling or tunneling is the process of non-covalent direct transfer of a reaction intermediate from the active site of one enzyme to the catalytic center of a second enzyme without prior dissociation into the bulk solvent. It is the channeling of different reactants between active sites, and the active sites can be located either on separate domains in a multifunctional enzyme or on separate subunits in a multi-enzyme complex. So, substrate channel is present in multi-enzyme complex as well as in the multifunctional enzyme. In this figure, you can see that E1, E2 and E3, these are three enzymes. They are closely associated with each other and they are forming multi-enzyme complex. In this case, you can see the substrate channel or metabolite channel here. Now, in this case, there is no, due to the presence of this substrate channel, there is no intermediate diffusion in the bulk solvent. And therefore, this substrate channel increases overall catalytic efficiency. Whereas in case of a soluble or dissociated multi-enzyme system, there is no substrate channel formation and therefore intermediates diffuse away in the bulk solvent and catalytic efficiency is decreased. There are many examples in a wide range of organisms, including eukaryotes and prokaryotes, where the assembly of different individual sequential enzymes of a metabolic pathway into functional multi-protein complexes increases the overall catalytic efficiency. In the case of primary metabolism, multi-enzyme complexes are involved in central carbon metabolism, including glycolysis, TCA cycle, fatty acid oxidation, Calvin cycle, amino acid biosynthesis, mainly tryptophan synthesis, the carboxysome and the proteasome. Metabolic reactions that are linked by multi-enzyme complexes have clear advantages over reactions that are catalyzed by individual or unlinked enzymes. There are advantages of multi-enzyme complexes due to the presence of substrate channeling. Substrate channeling has been proposed to decrease the transit time of reaction intermediates. Substrate channeling allows minimizing the distance that the product of a reaction must travel to reach the active site that catalyzes the subsequent step in the sequence, and being active sites close to each other in the multifunctional enzyme as well as in the multi-enzyme complexes. Substrate channeling has been proposed to prevent the loss of reaction intermediates by diffusion. The diffusion of substrates and products in the bulk solvent is minimized, then their dilution and decrease of concentration too. This leads to the production of high local concentrations, and even when their intracellular concentration is low. In turn, this leads to an increase in the frequency of enzyme substrate collisions. Therefore, substrate channeling has been proposed to protect labile reaction intermediates from solvent, to sequester reaction intermediates that are toxic to the cell, to circumvent unfavorable equilibria, to forestall the entrance of reaction intermediates into competing metabolic pathways, to prevent the buildup of excess reaction intermediates, and to closely regulate a block of consecutive reactions within a metabolic pathway or in a multi-step catalytic cycle. Now I'm going to give you a few examples of multi-enzyme complex. First of all, pyruvate dehydrogenase, I'm going to discuss about it in detail. Whereas 2-keto dehydrogenase, proteasome and others, I'm going to discuss about them in short. Pyruvate dehydrogenase complex is a classic example of multi-enzyme complex. This is the overall reaction where pyruvate is converted to acetyl-CoA and NAD plus is converted to NADH and carbon dioxide is released. Pyruvate dehydrogenase complex is a bridge between glycolysis and TCA cycle. Pyruvate dehydrogenase complex and enzymes of citric acid cycle are located in the matrix of mitochondria. Pyruvate freely diffuses through the outer membrane of mitochondria through the channels formed by transmembrane proteins. Pyruvate translocase is the protein embedded into the inner membrane which transports pyruvate from the intermembrane space into the matrix in symport with H plus and exchange, that means antiport, for OH minus. Pyruvate dehydrogenase complex is a multi-enzyme complex containing three enzymes, namely pyruvate dehydrogenase, dihydrolipoyl acetyl transferase, and dihydrolipoyl dehydrogenase. That means E1, E2, and E3. It has five coenzymes: TPP, NAD, FAD, H-square, and lipoamide. TPP is a prosthetic group of E1, lipoamide is a prosthetic group of E2, whereas FAD is a prosthetic group of E3. Pyruvate dehydrogenase complex is giant with molecular mass ranging from 4 to 10 million Daltons. Total 5 steps are involved for the conversion of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex. Already I told you that there are three enzymes present in pyruvate dehydrogenase complex, namely E1, that is pyruvate dehydrogenase, E2, that is dihydrolipoyl acetyl transferase, and E3, that is dihydrolipoyl dehydrogenase. All the 5 reactions are catalyzed by these three enzymes. First step is conversion. Pyruvate is decarboxylated by pyruvate dehydrogenase with help of TPP. Second step, the TPP is oxidized and transferred as the acetyl group to lipoamide. This forms hydroxyethyl TPP, and H plus ion is required for the intermediate to give off CO2. So by these two steps, CO2 is released. Third step, E2 oxidizes hydroxyethyl to acetyl and then transfers acetyl to CoA, forming acetyl-CoA, which is the final product of this reaction. Now, acetyl-CoA was made in the previous step. However, the process is incomplete. The E2 is still attached to the acetyl-CoA molecule, so E3 oxidizes the thiol groups of the dihydrolipoamide back to lipoamide. In the fifth step, that means that is the final step, as a side reaction, NAD plus becomes reduced to NADH. So from one molecule of NADH, 2.5 ATP is synthesized. So these are the 5 steps involved for the conversion of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex. Second example of multi-enzyme complex is alpha-ketoglutarate dehydrogenase complex or 2-keto-dehydrogenase complex or oxoglutarate dehydrogenase complex. The reaction catalyzed by this enzyme in the citric acid cycle is alpha-ketoglutarate plus NAD plus CoA. Those are converted to succinyl-CoA plus CO2 plus NADH. That means this is the multi-enzyme complex that catalyzes the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. This is a multi-enzyme complex just like pyruvate dehydrogenase complex. It consists of 3 protein subunits including E1, E2 and E3, that means oxoglutarate dehydrogenase, dihydrolipoyl succinyl transferase, dihydrolipoyl dehydrogenase. That means the oxoglutarate dehydrogenase complex has the same subunit structure like PDH and thus uses the same coenzymes as the pyruvate dehydrogenase complex. Those are thiamine pyrophosphate, that means TPP, lipoic acid, coenzyme A, FAD, and NAD. Third example of multi-enzyme complex is proteasome. Proteasomes are cylindrical multi-subunit proteases found in eukaryotes, eubacteria, and archaebacteria. It is involved in the ubiquitin proteasome pathway, control of cell cycle progression, in the termination of signal transduction cascades, and in the removal of mutant, damaged, and misfolded proteins. Already I have made separate video on this topic, that means on the topic of enzyme degradation in the proteasome, you should watch that video. Let's see a few other examples of multi-enzyme complex in a table. This table is showing few other examples of multi-enzyme complexes, their components, functions, and reactions. First of all, tryptophan synthase. This is one of the very important examples of multi-enzyme complexes, and the components are alpha to beta 2 complex, that means 2 alpha subunits and 2 beta subunits. The bifunctional tryptophan synthase alpha to beta 2 complex catalyzes the final two reactions in tryptophan biosynthesis. It channels a metabolic intermediate, that is the indole, between two active sites. So this enzyme is converting IGP, that is indole 3-glycerol phosphate, to L-tryptophan, and intermediate is the indole. Second example is carbamoyl phosphate synthase, which is a heterodimeric enzyme composed of a small and a large subunit. So components are alpha beta N terminal and beta C terminal. It catalyzes the ATP dependent synthesis of carbamoyl phosphate from glutamine or ammonia and bicarbonate. Third example is glutamine phosphoribosyl pyrophosphate aminotransferase, that is ATPase, and components are alpha beta 4, that means 4 alpha chain and 4 beta chains. It catalyzes the conversion of 5-phosphoribosyl 1-pyrophosphate, that means PRPP, into 5-phosphoribosyl 1-amine, that means PRA, using the amine group from a glutamine side chain. This is the committing step in de novo purine biosynthesis. Fourth example is cellulosomes. Two components are type 1 Dockerin linked enzymes and cohesion linked scaffolding proteins. These are two components, and it mostly extracellular cell surface cellulase complex, that means cellulosomes, that can proficiently hydrolyze the compact and recalcitrant cellulose microfibrils. That means cellulose is converted to monosaccharides, that is the glucose, by cellulosomes. For the further understanding of substrate channeling, you should read this mini review, namely the molecular basis of substrate channeling. And in this mini review, you can find few other examples of multi-enzyme complex, namely the dihydrofolate reductase thymidylate synthase. And in this review, you can see a very good clear picture of channel present in carbamoyl phosphate synthase. And in this paper you can find another example, that is the tryptophan synthase alpha to beta 2 complex, where in this multi-enzyme complex you can see tunnel present in this complex, and you must read this paper. Secondly, you should read this article, namely substrate channeling and substrate channeling molecular basis, where you can find the advantages of substrate channeling. These are given, and you can get a very clear picture of one enzyme, that is carbamoyl phosphate synthase, that is the multi-enzyme complex, where substrate is glutamine and product is carbamoyl phosphate. And in between that you can see inside the multi-enzyme complex blue colored substrate channeling or tunnel is substrate channel or tunnel is present, and it is clearly shown that this enzyme complex is composed of large subunit beta and small subunit alpha. So you should read this article also. And finally, you should read this paper, Engineering of Metabolic Pathways by Artificial Enzyme Channels. And here also you can find advantages of multi-enzyme complexes. These two figures are very important. Okay, so these three papers are important for the multi-enzyme complex. You should read those papers. I'm going to share links of these three papers in the description box.
I hope this video is helpful for you. Kindly like, share and subscribe this channel. Don't forget to press the bell icon for the further notification.